Hello everyone, welcome to Biochemistry Tutor. So in this video, we will be discussing certain basic facts about amino acids. So amino acids, from the name it suggests, it has an amino group and an acid group. Amino acids are the individual units of proteins. In other words, we can say that proteins are the polymers of amino acids, right? Amino acids are organic compounds containing two functional groups, an amino group and a carboxyl group. It is a typical structure of amino acid, where this one is the amino group and it is the acid group. Here the R group is variable. It varies based on the structure, size and charge. The amino group is the basic one, while the carboxyl group is acidic in nature. Amino acids have carboxyl group and amino group attached to the same carbon atom, which is known as alpha carbon. So this carbon atom is termed as the alpha carbon, where carboxyl group, amino group are attached to the same carbon, and the subsequent carbon atoms in this R group are termed as beta, gamma, delta and so on. They differ from each other on the basis of side chain, as we have already said. The standard amino acids have been assigned 3-letter abbreviation and 1-letter symbol. So there are 20 standard amino acids. They are assigned as 3-letter abbreviation or 1-letter symbol. This 1-letter symbol helps to reduce the size of data files which is used to describe an amino acid sequences. It also helps in easy memorization and understanding. Except glycine, all other standard amino acids are optically active, right? So why they are termed as optically active? Because the alpha carbon atom is attached to 4 different groups. So the alpha carbon atom is known as a chiral center and they are optically active. Whereas in case of glycine, which we will be discussing lately, the 4 carbon atoms, the carbon atom is not attached to the different groups. The absolute configuration of amino acids are based on the absolute configuration of 3-carbon sugar glyceraldehyde, of course. So this amino acids, the configuration of amino acids is based on the configuration of glyceraldehyde. This is the structure of L-glyceraldehyde and this one is L-amino acid. This is D-glyceraldehyde and D-amino acid. Here OH group is on the right hand side. Here this amino group is on the right hand side. So they are in the D configuration. Here OH group is on the left hand side and this amino group is on the right left hand side, so they are in L configuration. Okay. The amino acid residues are joined to each other by a special type of covalent bond called peptide bond. So we have already told, right, amino acids are the monomers of proteins. So here individual amino acids are joined with each other by a special type of covalent bond called peptide bond, and this peptide bond is formed by the removal of a water molecule, and that is the reason why amino acid, the term residues is used. This type of bond formation is an example of condensation reaction. It is formed by the removal of water molecule. The number of peptide bond is equal n-1. So this is the formula for finding out the number of peptide bond, where n it is the number of amino acids. It will be more clear when I give the example. So this is the dipeptide, formation of dipeptide. Here it is the first amino acid. This one is the second amino acid. Here a hydroxyl group from the COOH group is removed, and here hydrogen from the NH2 group is removed. So the water molecule has been removed here. So there are 2 amino acids, right? So the number of peptide bond will be 1. Why? Because n is the number of amino acids. So n-1, 2 minus 1 is 1. So the number of peptide bond is 1. And the amino acid formed is a dipeptide. So here it is the formation of tripeptide. Here it is the first amino acid, the second and the third one, and it shows the removal of water molecule from individual amino acids, and we will get a tripeptide. The reaction, like in the similar manner, we will get tetrapeptide, pentapeptide, hexapeptide and so on. So there are 20 standard amino acids. The first amino acid discovered was asparagine in 1806. The last of the 20 to be found was threonine, which was discovered in 1935. So then we can study the structure and the abbreviation and one letter code for the 20 standard amino acid. The first and the simplest one is glycine. Here the R group is replaced by hydrogen. Okay. It is L-glycine because the amino group is present on the left hand side, so it is present in the L stereoisomeric form. And the three-letter abbreviation is GLY, the first three letters, whereas the one-letter code is G. But it is not the case for all amino acids. There are certain amino acids where this three-letter abbreviation and one-letter code doesn't coincide with the name of the amino acid. This glycine is optically inactive, as I have already said, because there are two similar groups here, hydrogen and hydrogen. The second one is alanine. Here it is optically active, right? Because the four groups are different. It is L-alanine. The three-letter abbreviation is ALA, whereas one-letter code is A. The third one is valine. Here the group is a branched chain. The three-letter abbreviation is VAL, whereas the one-letter code is V. The fourth one is leucine. It is also a branched chain R group. And here the three-letter abbreviation is LEU, whereas the one-letter symbol is L. It also has the R group here is branched chain. Next one is isoleucine. Then here ILE. Look. Here it is ISO, whereas it doesn't coincide with this three-letter abbreviation ILE, but this one-letter symbol coincides with the first letter of the amino acid. Here the R group is branched chain. The sixth one is methionine. The three-letter abbreviation is MET, whereas one-letter code is M. Here the group is thioether group. This one is the thioether group. The seventh is proline. It has a pyrrolidine ring. This is termed as pyrrolidine ring. It has a secondary amino or the imino group present, which is NH2+. Of course, the three-letter abbreviation is PRO, whereas the one-letter symbol is P. The eighth is a phenylalanine. It is an aromatic amino acid, right? Here the three-letter abbreviation is PHE, whereas one-letter symbol is F, and the group is phenyl or the benzene group. The ninth is tyrosine. Here we can say it is a phenol group present or a hydroxyl group is present. TYR, and look, the one-letter symbol is different from the first letter of the amino acid, right? So it is Y, where this is T. Next is tryptophan. This is the indole group present here. Here the one-letter symbol is W. It is also different, whereas the three-letter abbreviation is also different, like TRP. Here it is TRY. So there are certain amino acids where the three-letter abbreviation and the one-letter code doesn't coincide with the first three letters of the amino acid. The 11th one is serine. Here hydroxyl group is present. This SER and S. 12th one is threonine. THR, T, hydroxyl group is present. The 13th one is cysteine. So, cysteine, here CYS and C. Sulfhydryl group, SH group is present. Here cysteine can form a dimer, which is also termed as cysteine, and there is a disulfide group present here. So, this disulfide bond can be formed between a polypeptide chain or different parts of the polypeptide chain. The 14th one is asparagine, which is the first amino acid discovered, right? And there is an amide group present. C double bond or NH2, then ASN and N is the symbol. Next one is glutamine. Here if we add CH2 group to the previous one, then we will get glutamine. GLUQ amide group is present. The next one is arginine, which is ARGR, guanidino group is present. The 17th one is lysine. Here the epsilon amino group is present because the carbon atoms in the R group are designated as beta, gamma, delta and epsilon, and this amino group is attached to this epsilon carbon. So we can say that epsilon amino group is present here. The 18th one is histidine. So here imidazole group is present. The 3-letter abbreviation is HIS, whereas 1-letter code is H. The 19th one is aspartate. So L-aspartate, ASPD, beta-carboxyl is present. So this aspartate and the next one can be termed as the amides of asparagine and glutamine. So if you can recall the structure of asparagine and glutamine, so this instead of this COOH, there is C double bond O NH2 present. Amide group is present. The 20th and the last standard amino acid is L-glutamate, GLUE. So here gamma carboxyl is present. So beta, gamma, and yeah. So in recent years, a 21st amino acid is added, which is known as selenocysteine. It is an unusual amino acid containing element selenium in place of sulfur atom of cysteine. So this is the structure of cysteine. Here in this is a sulfhydryl group. Instead of sulfur, we will get we have this selenium atom attached to it. So this selenium or the selenocysteine, it is found at the active site of certain enzymes like glutathione peroxidase, glycine reductase, and also present in certain proteins like selenoproteins. This incorporation of selenocysteine into the protein during translation is carried out by the codon, namely UGA. It is a stop codon, right? So this UGA help in incorporation of selenocysteine into the protein during translation process. So there is also a 22nd amino acid, which is pyrolysin. Here this stop codon UAG code for pyrolysin. So as far now we have only 20 standard amino acids and we have discussed their structure. And in recent years, 21st and 22nd amino acids have been added. And the next category is uncommon amino acids or the rare amino acids. What are those? 4-hydroxyproline. So of course it is a derivative of proline, and it is present in plant cell wall protein and also in collagen, which is a fibrous protein of connective tissue. The next one is 5-hydroxylysine. Yeah, it is a derivative of lysine and it is also present in collagen. The next one is 6-methyl lysine, and it is a constituent of myosin. This myosin, it is a protein which is present in the contractile system of skeletal muscle. It helps in the contraction of the skeletal muscles. The next one is gamma carboxyglutamate, which is found in the blood clotting protein prothrombin. The next one is desmosin, which is a derivative of 4 lysine residues. It is found in elastin. Okay. So amino acids act as acids and bases. So due to the presence of this ionizable R group, amino and carboxyl group, it can function as weak acid and bases. Substances having this nature are amphoteric, and hence amino acids can be termed as ampholyte. A term zwitterion or dipolar ion is a hybrid molecule. So zwitterion is the molecule which contains positive and negative ionic group. It is derived from the Greek word which means hybrid. So this amino acid can be, it is mostly present in the zwitterionic form, and it is rarely present in the neutral form. This one is a neutral form and it is the zwitterionic form. In biological system, amino acid is mostly found in this form, zwitterionic form. So this neutral amino acid eliminates one H plus ion and it will form anion. So in high pH, this amino acid releases a proton and it exists in anionic stage. In low pH or in the acidic stage, it will accept one H plus ion and it is present in the cationic stage. So to form zwitterion, here this cation has to remove one H plus and this anion has to accept one H plus, so we will get this zwitterion. Each amino acid has a characteristic pH where it is present in the zwitterionic form, and that characteristic pH is termed as the isoelectric pH, which is denoted by Pi. So isoelectric pH is nothing but the pH at which the amino acid is present in its zwitterionic form. So yeah, that's it guys. Thank you very much. I hope you understand something and you gained some knowledge from this video. I will be uploading along with this how to draw the structure of amino acid. So stay tuned for more videos.